The definition for epigenetics is defined as the study of how cells and organisms change gene expression and function without changing the base sequence of DNA. So there are three different ways on the molecular level in which we can alter gene expression. So one of the first methods is to modify DNA through methylation process. And what that means is to add a group, a chemical group, CH3, onto the promoter region where cytosines are located. And by having a cytosine being having the CH3 bounded onto the cytosines would actually prevent transcription from occurring. However, what that also implies is that if you can remove methyl groups on a gene that you do not want to express, that can also cause problems, like in the case of cancer diseases. Another method in which this can occur is by changing how tightly coiled the DNAs are with the presence of histones. So if there are presence of a lot more histones being there, then the DNA would be more tightly coiled, in which case you cannot unwound the DNA in order to read the bases that are paired together inside the double helix. And last but not least, you can also change transcription and translation with non-coding or regulatory regions of the RNA. So let's take a look at what I mean by that. All right. In essence, in part A of this diagram, what you're looking at is a nucleotide. So here is the deoxyribose. Here is the base itself. In this case, this is cytosine. And by adding the methyl group CH3, you will notice that this is where the binding occurs. So this process is what is known as methylation process. Then if you take a look at diagram B, what it is showing here is a regular double, this is a double helix, and where you see the placement of this vertical line, it represents unmethylated sites along the areas where you can find cytosine. If the areas are unmethylated, transcription would occur, as is indicated by this arrow down here. However, if you start adding this orange dot, which is representative of the CH3, the methyl group, along the areas where cytosine is positioned, then what happens is gene expression would be inhibited in this promoter region of the operon that you have learned so far. Now, the second method that was mentioned in this particular slide about modifying the chromosomal proteins with the use of histones, this is an interesting one too. So all of the genetic material are generally coiled around histones, and they are represented by this light green dot that you see right here. All right. The more of these light green dots you have, the more tightly coiled DNA becomes. So the more tightly bound that they are, therefore reading the information that is coded in the DNA becomes more challenging. So therefore transcription can be hindered as a result of that. Now, under normal circumstances, the transcription process will flow, as is indicated here. So each one of these purple dots is representative of the transcription process, the RNA polymerase moving along the way, copying, making messages that are either rRNA, transfer RNA, or messenger RNA along the sequence. However, there's another way in which you can alter the expression of the transcription translation process. If you use an acetyl group, which is another chemical group that is indicated on this slide, which is COCH3, and you add that onto the histone itself, that can actually help loosen the winding of the DNA, the uncoiling process itself. Because you're able to loosen it, transcription will become more active. And the term that we use is called hyperacetylated chromatin. Remember, chromatin is when DNA is stretched out, not in this tightly coiled form that we sometimes where we call it a chromosome. Now, on the other hand, if you think about it, right? If you hypoacetylate it, meaning you reduce or you remove the number of acetyl groups that are here, that would actually help the chromosome to become more tightly coiled. And then on top of it, if you were to add the methyl group with the help of methylase, not only would this DNA be more tightly wound, you will notice that you see the distance, there's almost no gap, very, very little gap to no gap between these genetic material, whereas in the earlier portion of this diagram, there is a significant gap right here. Hopefully you can see where my cursor is pointing at, all right? You will have the RNA polymerase will have a much harder time trying to transcribe this area. As a matter of fact, the combined effect can actually be used to silence a gene, stopping the process of transcription. Now, having this background knowledge, what I want you to do as part of your exit challenge is to read carefully page 227. On this particular page, there is a textbook box where you can read a little bit more about two scenarios that are really quite interesting. What I want you to do is to create your own set of diagrams that can illustrate what the textbook would describe in words. How is the insulin gene regulated during early embryonic development stages compared to later in life? Now, that actually is described in paragraph 2 of this page. And the second scenario that I would like you to make a diagram for me is how the methylation process is related to the onset of Angelman syndrome, which you have learned during chapter 10, if you remember. One of the things that you learned is about genomic imprinting. So this is where it comes back again, all right? But now with the added knowledge of how methylation process occurs, I want you to add captions to your diagram. And I want you to keep in mind that the goal of this mini activity where you create your own diagram is to formulate connections between what you have learned in this lecture, specifically about methylation process, acetylation, and the presence of histones, how you can connect it to the previous knowledge that you have gained in the earliest chapters. Now, what I will be including at the bottom of my video lectures on YouTube are the following review videos that you can watch on your own at your own time that goes over gene regulation produced by the Amoeba sisters. You can also go over the Bozeman science video regarding Operons on your own if you find these to be helpful. Try also this one about epigenetics, which is really fascinating. See which one of these you are comfortable with if you think it's necessary for you to learn the material, okay? And this is the end of the lecture. This is a slightly shorter one. I look forward to receiving your diagrams and taking a look at it, all right? Let me know if you have any questions regarding those diagrams, okay? Take care. See you soon. Bye-bye.